All right, welcome back to episode 5 of the MCAT prep series. And today we're going to be continuing the biochem section by talking more about protein structure and in specific conformational stability. And so by the end of today's video, you should be able to understand protein conformational and native stability, understand the forces that maintain protein stability, be able to explain protein folding and denaturing, and understand the hydrophobic effect and the salvation layer principle. So moving into content, the first thing that we're going to be looking at is conformational stability. And what is this? Well, it's how well a protein maintains its 3D native structure under physiological conditions. And so the highlight, or the most important part of this definition, is this word maintains. So conformational stability can essentially be thought of as how strongly proteins resist change structurally. And so, if you take a look at this protein, which I'm going to put on screen now, you'll see that there are constantly small structural fluctuations that are happening all the time. And this is true for every protein in the body or every protein in any system. And so one important thing to keep in mind is that while these structural fluctuations are occurring, the functional component of the protein is not lost. And so conformational stability is how well a protein can resist change in a way that maintains its function. And so one more thing to keep in mind is that this conformational stability isn't really, magic. It isn't really the protein just deciding I want to be folded in this manner one day and folded in this manner another day. It really has to do with the free energy of the protein, and specifically the free energy difference between the protein when it is folded and when it is unfolded. And so if this idea of free energy is a little bit confusing now, it is something I'm going to go into in greater detail in future videos, but essentially it can be thought of as the driving force for everything. So free energy determines which direction reactions go in, it determines equilibrium, and in this case, it determines how proteins are folded. And so proteins will tend to fold in a way that reduces or minimizes their free energy. Okay, so moving on, the next thing we're going to be talking about is native conformation. And so what native conformation is it's the functional 3D structure of a protein under physiological conditions. So whereas conformational stability is how well a protein resists structural change, native conformation is the actual physical 3D structure of that protein. And so one thing I just want to highlight in this definition is that it has to be functional. And so when we're thinking about native conformation and when we're thinking about how proteins are folding and which secondary, tertiary, and quaternary structures are being created, we have to keep in mind that at the end of the day, the protein that we're creating still has to be functional. And so you might be asking yourself, it feels sort of random, or how might proteins know how to fold or which structures to create to find that native conformation? And so really what's important here, just like conformational stability, is free energy. Because in order to create a native conformation, we're looking for the lowest free energy structure possible that still has function. And so we're trying to minimize free energy with our native conformation. And so again, you might be asking yourself, how is it possible? How is it possible that creating an alpha helix in one location or a beta sheet in one location minimizes free energy? So that is, how do proteins know how to fold to minimize energy? And so in order to think about this, we need to consider this equation of delta G equals delta H minus T delta S. And so you may be familiar with this equation, but if you're not, just to go over the variables, delta G is our free energy, delta H is our enthalpy, T is our temperature, and delta S is our entropy or change in entropy. And so the whole point of a native conformation and when proteins are being created is to get this free energy term as low as possible. And so keeping that in mind, let's take a look at this diagram which I've created to illustrate this principle. So let's pretend that this squiggly line at the top is a protein in primary structure, and this one down here is a protein in secondary structure. And so in the process of going from primary to secondary structure, we're creating, let's say, a beta sheet, for example, and some bonding is going on. Well, it turns out that this addition of bonds, or this creation of bonds, actually lowers our enthalpy. In addition, it also lowers our entropy. And so now if we take a look at the equation, delta G equals delta H minus T delta S, we're seeing that this value, delta H, is becoming more negative, and this value, delta S, is becoming more negative. And so now you might be asking yourself, if our goal is to get delta G more negative, but we're subtracting a more negative delta S, aren't these two just going to cancel out? And yes, that is the case. However, one thing that I didn't mention now, but we will go into later in this video, is that whereas the delta S of the protein is being decreased because of bonds being formed, because there is less randomness, and delta S can be thought of as a measure of randomness. So because you're creating more order, there is lower entropy between the protein. However, this lower entropy between the protein is also paired with an increase in entropy of surrounding water molecules. And again, we'll go into that later in the video. And so now we can see that although we are losing entropy, we are also gaining it through water molecules. These cancel out, and overall, our delta G is going lower. And so that is how the creation of secondary structures or the creation of tertiary quaternary structures might lower the free energy of a protein, simply just by creating bonds. And so moving on, we're just going to do a quick recap from content that was actually covered in the last video about certain intermolecular forces, like the ones I've listed below, that are responsible for stabilizing secondary, tertiary, and quaternary structures in proteins. And so just remember that covalent interactions, an example of them are disulfide bonds, which we've talked about in length. An example of electrostatic interactions can be salt bridges. So for example, interactions between a positively charged side chain and a negatively charged side chain. We also have hydrogen bonds, which can be in between the protein backbone in the case of alpha helices or beta sheets, or between side chains, which are those R groups, hydrophobic interactions, which we will go into later in this video, and then van der Waals interactions, which remember are individually weak, but because there are so many of them, become strong collectively. The reason I starred the hydrophobic interactions is because it actually turns out that this single interaction is the largest driving force of protein structure. So you can think of it as hydrophobic interactions really play a big part in how proteins end up folding or how they end up creating themselves in 3D space. And so now let's think about that. So protein folding is the process by which a linear polypeptide, so something in primary structure, turns into its native 3D structure. So that is protein folding. And, the way I've spoken about it in this video sort of suggests that proteins do this on their own, but it's actually the case that proteins are assisted in this process by molecules called chaperones, which again, we'll speak about in later videos. And essentially what you can think of chaperones as doing are, like their name suggests, helping proteins go from their primary linear structure into their complicated 3D structure. And so they prevent aggregation, for example, which means they prevent proteins from, clumping together and forming a sort of mess. They provide isolated environments for the process of protein folding to take place, and they increase folding efficiency overall. And so when we're thinking about protein folding, one of the biggest or most important concepts from this entire video that you can keep in mind is that primary structure determines secondary, tertiary, and quaternary structure. That is to say, primary structure determines native structure of a protein. And so when we're thinking about this idea, one of the key experiments to keep in mind is the Atkinson experiment. And so this experiment is really one that proved that primary structure determines native or 3D structure. And so how this experiment worked is that the protein, this one, ribonuclease A, was exposed to urea, which destroyed H bonds or hydrogen bonds, and beta-mercaptoethanol, which destroyed disulfide bonds. And so, as you might imagine, by destroying hydrogen bonds and by destroying disulfide bonds, the protein ribonuclease A unfolded. But now here came the interesting part and where the most significant findings of the experiment stem from. Once both chemicals were removed, so once you removed the urea and the beta-mercaptoethanol, the protein spontaneously refolded. And you're back to the native structure. And so, this at the time was an extremely significant revelation. You take a protein that's fully functional, it has the right structure, add some chemicals, it unfolds, which is what you might expect. But then when you remove the chemicals, the protein somehow goes back to the structure it was in the first place. And so what does this show us? Well, this shows us that the primary structure, which was the only structure that was unaffected by the chemicals, determines the 3D or native structure of the protein. because somehow all the information that was required for the protein to fold into its correct orientation was inside of that primary structure. And so that's an extremely high yield topic that most definitely will come up on the MCAT. Okay, so now let's move on to denaturation. So this whole time for all of last video and all of today's video so far, we've been talking about how proteins build themselves up, how they go from a primary structure into a secondary structure, tertiary, quaternary, etc. And so when we're thinking about biochemistry, not only is it important to understand how proteins fold and how they come to be in their native conformations, but it's also really important to consider how denaturation works, or how we can take a protein that's in its native structure and unfold it, how we can bring it back to its primary structure. Because denaturation and loss of native structure also eliminates protein function. And so, for example, if we want to see what a protein does in a cell, we can denature it, make it lose its function, and then see what happens. And that will give us, as scientists, a clue as to what the original structure of the protein may have been. And so when we're thinking about denaturation, we need to remember that primary structure always is going to stay intact. So we might be removing secondary structure, we might be removing tertiary structure, we might be even removing quaternary structure, but the primary structure of our protein will stay undisrupted. And so there are a few ways to denature proteins. And when we're thinking about denaturation, we need to be thinking about methods that are going to get rid of bonds, because essentially that is how you denature a protein. All the secondary and tertiary and quaternary structures we looked at rely on bonds, whether they're covalent, whether they're non-covalent, ionic, etc. Bonds are what stabilizes a protein structure. And so certain methods that destroy bonds and in turn destroy protein structure can be heat. It can be changes in pH, either making it lower or higher, or reducing agents. And while there are a lot more ways to disrupt protein structure, these are some of the more common ways that you might see on the MCAT. And at the end of the day, you just need to remember that these methods of denaturation, and denaturation in general, is just a way of removing bonds. And then one more thing to keep in mind about denaturation, and one way that this test might possibly trick you, is, again, going back to this fact that primary structure stays intact. So denaturation of a protein does not equal hydrolysis. So it does not equal hydrolysis. And so that means that we're not breaking peptide bonds, and that primary structure of our protein, no matter how much we denature it, it's always going to stay intact. And so moving on, we're going to be talking about hydrophobic interactions and solvation layers. And now if you get anything from this video, it really should be from this topic, because this topic is easily one of the most highly tested concepts from all the topics I've given in today's video. And so you might be under the misconception that hydrophobic molecules simply just attract each other. But this is not the case. And one thing I'm really going to try to show you with this next demonstration is that hydrophobic molecules don't attract each other, but instead the hydrophobic interaction between them is driven by water. To demonstrate this principle, let's pretend we have valine, like I've drawn here. And valine is just floating around in solution, surrounded by water molecules. So if we look at these water molecules surrounding the valine, we'll notice that they can't really interact with the valine. Remember, these oxygens have a partially negative charge, and these hydrogens have a partially positive charge. But valine is a hydrocarbon, it's nonpolar, it's completely uncharged. And so it doesn't want to interact with water. And so if the water can't interact with the valine, well then what does it interact with? Well, it's going to have to interact with other water molecules nearby. And so this is really the foundation of the hydrophobic effect. And it's that water molecules surrounding a nonpolar group become highly ordered. They want to maintain a hydrogen bonding network between each other. And so they really are limited in movement. They become extremely highly ordered around a nonpolar group in a way that maximizes their interactions with themselves and minimizes their interactions with the group they're surrounding. And so this ordered shell is what can be known as a water cage, for example. And it's just one way to think about it, where the water has really put this nonpolar group in a cage. And so if you compare the waters, in a structure like this to the waters in a structure like this, you'll notice that we have lost a lot of freedom when it comes to how the water molecules can move or how they can orient themselves. And so as a result, this loss of freedom, or this loss of randomness, is correlated with a decrease in entropy. And so let's just keep that in mind as we keep going through this example. So now let's pretend that instead of one valine, we have two valines. Well, there are two main possible ways that we can orient them in our example. We can put the valines on top of each other, like I've drawn here, or we can put the values sort of far away from each other in solution. And so taking a look at both of these possible ways that we can orient the values, we see that in method 1, which I'll just call this one up here, we just have one main water cage. But in method 2, which I've drawn down here, we have two water cages. And so you might be asking yourself, okay, so what does it matter? Whether we have one water cage, two water cage, who cares? Well, like I said, the formation of a water cage decreases entropy. And we know because of the equation delta G equals delta H minus T delta S, that we always want to be decreasing delta G, our free energy. And so that means we don't want to be decreasing our entropy. We want to be increasing it since it's a negative term. And so when we're looking at, whether we want to orient the values like this or the values like this, what we need to be thinking about is we need to be thinking about this situation from an entropic perspective. Which scenario allows more freedom? Which scenario allows less water molecules to be trapped? And that is scenario A. So this one up here. Because if we look down here in scenario B, we're seeing that we have way more water molecules that are oriented in these water cages, that are stuck and can't move. And so that is essentially the driving force between the hydrophobic effect. It's an effect between nonpolar groups that minimizes the amount of water in water cages, and we're maximizing the entropy of free water molecules that aren't involved in water cages. And so although the protein itself becomes more ordered because we're putting those non-covalent groups all in one area, which decreases entropy, the increase in entropy that's coming from water molecules that now don't have to be in water cages is even greater, and is actually the driving force for this effect. And so, just to recap, the main conclusion from this demonstration is that the hydrophobic effect is driven primarily by the entropy of water, not by the hydrophobic groups attracting one another. And if you can take anything away from this video, it's really that idea, because I can guarantee you it will be tested on in some way. And that's it for today's video. So if you have any questions, please feel free to leave them in the comments. I know this one could be a little bit more confusing to understand initially, and good luck with your studying.